Key Assessment 2, Question 3. This is Spirogyra. If you've seen the colour photo of this, you'll notice that these are green blobs. And then there's a kind of greyish blob there. It tells you it's autotrophic, which means it does photosynthesis. Identify the organelles. A big blob in a cell. Well, probably a nucleus. B, these green blobs, or lots of them, but it tells you it's autotrophic. So these are chloroplasts. A magnification calculation, hopefully you've done loads of these. The information we've got, XY, there is 32.3. So our actual size of XY is 32.3 micrometers. Our image size, well, we need a ruler on there, and when we get our ruler on, we measure 12 millimeters. So XY is 12 millimeters. When we do a calculation, we need to use the same units, so it's easier to convert my millimeters into micrometers. 12,000 micrometers. Magnification is how many of my actuals go into my image. So my image divided by my actual. My image is 12,000, my actual is 32.3. 12,000 divided by 32.3, 371.51, 371.517. So I'm going to round that up to a sensible number, 372 times. During the day, concentration of solutes, so we're going to talk about transport and osmosis, that sort of thing. But the cells remain almost constant in size. What do you conclude about how the structure of the wall enables Spirogyra to survive? Now, a bit of a strange one, this one. So 4 marks. We need to talk about the structure of the cell wall. So it's a plant, autotroph. Structure of the cell wall. We know that's strong. So cell wall provides strength, rigidity, that sort of thing. So that's our first point we're making. We can say another mark why it's strong. We can say, made from cellulose microfibrils, which are cross-linked with hydrogen bonds due to the monomers being 180 degrees to each other. I'll put more information there we need, but this sort of idea of those cellulose microfibrils from your knowledge. Then we need to say another 2 things, 4 marks. So we do need to say another 2 things. So we need to say, actually answer the question, how do we survive in different solute concentrations? So, for example, e.g., when we've got this evaporation it talks about, so high solute concentration equals low water potential. So water moves in to the cell by osmosis. So that's a mark there, and actually it's almost impossible. Due to me putting too much in there in that second point, I've run out of space. And so the cell wall prevents bursting. Or you might have made the reverse argument of water moving out if it, before the evaporation or when it rains, and the cell wall would prevent it from shrinking. And lastly, we've got a photomicrograph, so a light microscope image of the bacterium nostoc. Gives you a bit more information. It says that we're grouping these separately. A couple of jobs that we need to do. Conclude what cell types are present. So we need our cell structure information. And then it says 2 differences and 1 similarity. So cell types. So we're having Spirogyra is eukaryotic. Nostoc is prokaryotic. Those are the types of cells. A similarity, and we need 2 differences. So we've had 1 mark so far, so we need so let's do it in order. So 2 differences, lots of things we could say here. For example, we need to be clear which one we're talking about. Spirogyra has linear DNA. Nostoc, loop or circular DNA. Another difference, Spirogyra has larger ribosomes than Nostoc. Other things we could say, Spirogyra has got membrane-bound organelles, for example mitochondria, whereas Nostoc doesn't. The DNA is enclosed within a nuclear membrane, and so on. So just stuff from your table, from your notes. Then we need, so that's a mark if you get 2 of them, and then we need the similarity. So similarity, for example, both contain ribosomes. Both contain ribosomes. Both contain, so that'll get us the mark. We could have a membrane. Both contain DNA. So 1 of those similarities. 11 marks.